So we've talked about functions. We've talked about graded potentials, which can lead to an action potential. And we've talked about how an action potential speed can be affected. Okay, and so now we've basically got all the way down to the axon terminal. And it's at the axon terminal where a neurotransmitter or chemical can be released, telling another cell or another neuron to do something or to not do something. And that's what this video is going to talk about. And so with that, we've got these 4 topics, or these 4 outcomes. So we've got a synapse. And so a synapse is just a junction between a neuron and another cell. That other cell can be a neuron, but it can be your muscle or your heart or something else. So with the synapse, there are 2 different types. You've got an electrical synapse. This is where these cells are actually connected. It's a direct connection. And so when one cell becomes depolarized, they all become depolarized. This is something that occurs in your heart, and we will talk about more in the cardiovascular system. Then we've got the chemical synapse. And so this is where a neurotransmitter, a chemical, is released by what we call the presynaptic neuron, so the neuron before the synapse. Okay, and then this neurotransmitter will travel across that gap or that junction and will bind to a receptor on a postsynaptic cell. So if we look at this diagram here, here's our presynaptic neuron. These dots represent neurotransmitter or that chemical, and then you have your postsynaptic, in this case, neuron. Okay, and this is the process that we're going to talk about today. So neurotransmitters are just chemical signals that are released because an action potential reaches the axon terminal. Okay, these neurotransmitters will target a specific cell. Neurons are very directed. Okay, whereas a hormone that we'll talk about the endocrine system, those have more global effects and they're not as specific. Okay, neurotransmitters they occur rapidly, but their effects are short-lasting. Examples are acetylcholine and norepinephrine. Okay, now when it comes to neurotransmitters, you can classify them, or they can have what are called agonists, they can have what are called antagonists. Okay, there's a reason here that I've got a picture of Taylor Swift and Selena. And so neurotransmitters, agonists are neurotransmitters that bind to the same receptors. Okay, but what makes them agonists is not that they can bind, but they both activate that receptor. Okay, and so this causes the ligand-gated channel, okay, remember that's a chemically gated ion channel, to open. So it doesn't matter if it's acetylcholine, if it's an agonist of acetylcholine, it will open that receptor as well. Then we've got antagonists. And so again, antagonists are similar to agonists in that it's a neurotransmitter that can bind to the same receptor. But again, there's a reason that I've got Kanye and Taylor on this picture, right? They're not agonists. They're not friends. They don't help each other out. They don't really seem to like each other. Okay? And so in this case, an antagonist is a chemical that will bind to that receptor, but it will prevent the activation of the receptor. And as long as that antagonist is bound to the receptor, that channel will not open, so you have to get rid of it. Okay, so the ligand-gated ion channel remains closed. Okay, agonists, your friends, help you out. Antagonists are your enemies, not going to help you out. So here we've got a chemical synapse, and so there is a lot going on here, but as I said earlier, here's your presynaptic neuron, here's your postsynaptic neuron. We've got voltage-gated calcium channels that we have not talked about. We've got vesicles, which we talked about in the transport mechanism video, okay? And then we've got our postsynaptic neuron. And so we're going to talk about the process that occurs when an action potential comes down, ultimately leading to the activation of those channels there at the bottom. So how does this work? Remember that physiology is very mechanistic, meaning that one thing leads to another, leads to another, leads to another. Okay, so we have an action potential that arrives at the axon terminal, or the end of the axon, okay? This causes voltage-gated calcium channels to open. So they're shown here, right here, okay? But we've got them all over. So what happens? Well, we open a calcium channel, and you might remember from the graded potentials video, you might not, and that's okay. There is more calcium outside of the cell than inside. Well, since there's more outside than inside, this allows for calcium influx. So calcium will go into the cell. Okay. Calcium will then bind to these vesicles that are in here. Well, when that happens, exocytosis occurs. And when exocytosis occurs, these vesicles will move to the membrane, and ultimately they will release their neurotransmitter. This neurotransmitter will diffuse across the synapse where it will ultimately bind to a receptor. Now, we want neurotransmitter to be released from the neuron, but we ultimately need to get rid of this neurotransmitter as well. Okay? And the reason being is you don't want a cell to be continuously excited or inhibited. There are times where you want both. Okay? And so we need to get rid of this neurotransmitter, right? I don't want my bicep to constantly be contracted, otherwise I'm going to walk around like this. Not a great look. And so with this, we've got several different ways that we can get rid of a neurotransmitter. Okay, so one way is that neurotransmitter can just diffuse away in the bloodstream. Okay, so it's getting away from the cell that it was targeting, nothing's going to happen. Okay, it can be broken down by an enzyme. I refer to enzymes, have referred, and will continue to refer to enzymes as hammers. Hammers break stuff, right? If I give my 7-year-old a hammer, he's going to break stuff. It's just going to happen, so I shouldn't give him a hammer. Then we can also just take this neurotransmitter back up into the cell from which it came. Okay, so we can have reuptake, or it can just be taken up by another cell. So in this drawing, we've just got glial cell. It doesn't have to be a glial cell. It can be another cell. Okay, but here are 4 different ways that we can remove that neurotransmitter so that I'm not constantly walking around with my right bicep contracted. And so can you do these 3 things? Can you define neurotransmitter? Can you explain the difference between an agonist and an antagonist? And can you go through the steps that talk about how action potential is communicated from one neuron to another neuron at a synapse?